Okay, it's 12 o'clock, so we're going to get started. This is a really great presentation because I know because I've sat through it once, and I'm looking forward to picking up a lot of new information even today, watching it again. So I'm Ken Baker. I'm the interim director of the Idaho Integrated Design Lab, University of Idaho. On behalf of Idaho Power, our sponsor, and the IDL, I'd like to thank you for attending this session. We have a great topic and a great speaker today, Kevin Den Wymelenberg, whom I think most of you probably know. He was the first and a longtime director of this lab. He's now in Oregon at the Energy Studies and Buildings. Actually, I think he has lots of directorships at this point in time. I'm a little confused myself, but he is Dr. Den Wymelenberg, and I'll say more in just a few minutes. I want to go through a little bit of some logisticals. I think the way this is set up, most of you are muted now, and we do want to take questions at some point. So we'll work with you to unmute, and I'd say use the chat to start with if you have a question, and then we'll go to you and unmute you. I also want you to know that you can get CEUs for this session, and it's, I believe, 1 CEU. In order to get that, because of the logistical piece, I thought it's just easier if you email us, and Lindsay Watkins, lwatkins@uidaho.edu. That's at the bottom of this first slide, you can see. So if you're sitting out there in the audience watching and you would like to get a CEU, please email Lindsay and we'll see that that happens for you. And the lab is starting to do, I think, pretty well. Even with the COVID, things have changed a little bit on how we do business. Obviously, we're doing this webinar right now through a Zoom. Of course, with Kevin being over in Oregon, we probably would have done it this way. Yeah, anyway, there might have been a few in the classroom. But we have 3 full-time employees here now, myself and Damon and Dylan, and I think probably most of you know us. And we have 4 really great interns right now, research assistants. What they are, they're getting master's degrees, and 2 of them in engineering. Actually, Juven on the far left already has his degree, but he's going to come back and join us for a while.
Hey, and, Ken,
yes.
At least the feed I'm seeing is still stuck on slide 1, just so you know.
Oh, is it? Oh, mine's not. Okay. Huh. Tell me if it changes.
Not yet. I can see the PowerPoint window, so I see where where slide 2 should be. But yeah, it's not advancing. Maybe I'm the only one.
Okay. Well, if that's the case, I won't bother going through the slides. But I'll stop this share, and I'll just kind of walk you through this. Must be having some technical difficulties. So we have 4 great interns right now, 2 architectural and 2 engineering, getting their master's degrees. What I really want to tell you about is we have a lot of programs through Idaho Power Company. I told you earlier they're the sponsor of this session, and we thank them for that sponsorship. It's been a very long-term relationship we've had with them. There's programs you can find on the Idaho Power website, which is not in my head for what that is right now, but we can get that for you. And the Integrated Design Lab, we're able to provide technical assistance to you, directly to you, on projects that are energy related. So if you have a new design or an existing building that you want to work on, whether you're an architect or an engineer, we're happy to work with you. And up to, it depends on the level of assistance, but we can go up to $4,000 on level 3, where Idaho Power is actually paying our time, and we're cheap university people, so that's a lot of time. They're paying our time to actually provide you technical assistance, and we can do things like anything from massing studies to advising you on energy models, doing some daylighting work, all kinds of technical energy issues that we can work on directly with you. And I think most of you out there know about that at this point in time, because we've been around doing this for a while. But thank you, Idaho Power, for that. We also have a energy resources library. It used to be called the tool loan library. Lots of great tools. We have some books right now that we just cataloged. Gary Christensen just brought in a lot of his books and gave to us so that we can lend them to you. And then we have thousands of dollars worth of very nice monitoring tools that Idaho Power has purchased for us over the years. And we're happy to show you how to use those. There's an online resource on how you can check those out at idluidaho.com. And Dylan's just finished putting together a really nice tutorial and on site for that. So please use those tools. And we have other resources. And I'll stop now because I want Kevin to get most of the time here. But thank you again, Idaho Power. We really appreciate it. And now, Kevin, since my computer.
All right.
There you go.
I think we're good. You all can hear me and see the slide?
So let me just say, Kevin, we really appreciate your being here. And if I can stop my video of myself, I will. And Kevin van Den Wymelenberg, as I said, was the first director of this lab. And this research he's going to talk about today is really, it's state of the art, but he's been doing it for a while. This is what he's been doing in Oregon. So thank you, Kevin, and go ahead. And I'll
thanks.
Turn off
well, first of all, thank you, Ken. Thanks to the University of Idaho Integrated Design Lab. It was my home for 11 years and a very near and dear to my heart still today. So fun to see so many names. And I won't name them all because I'm sure there's actually quite a lot of participants today. So that's super exciting. But you know who you are, and thank you. It's fun to see your name, if not your face again. And also thanks to Idaho Power, as always, for their long-term commitment to leadership and energy efficiency and market transformation and support for research and innovation through the University of Idaho relationship. It was valuable and it continues to be valuable. And so thank you to Idaho Power. Really, I always said it, and I think it's still true. I'll still say it. The relationship with Idaho Power has been a really meaningful and impactful piece of my life. And I will also say is unique in the world as far as I've seen in terms of their commitment to this kind of research and practice. So kudos. Here I am again. More importantly, here's a group that has done a lot of the work that you're going to see today. In particular, Leslie in the blue sweater in the front row and Patrick to her left have been instrumental. And a quick shout out to Charlie Brown, my predecessor here at the University of Oregon, just passed away this February. If you weren't aware of that, I wanted to share that. But I really enjoyed this picture, which was not that long before he passed away. He lived a really long and full life and was just incredibly productive. And right up till the end, he worked till 2 months before he passed. A little bit about the Institute. There's some functions within the Institute that seem that are fairly similar, and certainly I learned a lot with my time at the Integrated Design Lab and trying to incorporate those kind of concepts. The Energy Studies in Buildings Lab is what I was brought here to lead, and then quickly was asked to take over the Biology in the Built Environment Center. And because of that integration, decided that there was really this interesting opportunity to try to transform what it means when we say healthy buildings. And so much of it is talk. And I felt like we were in a really unique position to bring some really great evidence to support what it means to be a healthy indoor space with the microbiome work at the Biology in the Built Environment Center. And then quickly built a partnership with the other lab here, the Baker Lighting Lab, with a new director that had just taken it over. And then Allison Kwok, her name is Siobhan Rockcastle, and then Allison Kwok is leading the Net Zero Energy Case Studies Lab. We also have some great partnerships with the Tallwood Design Institute focused on wood, and then just a whole range of partnerships with other disciplinary researchers from other universities, as you can see in the top left corner. And then on the bottom right, a pretty strong industry engagement where these individuals on the bottom, on the right side, are all paying a subscription fee, and we collaborate together to ask tough questions that are facing practice and develop seed research projects that the students work on to try to begin to answer those questions and really be high risk exploratory kind of work that then can lead to more successful federal funding opportunities to try to bring that back into the fold. And so as Ken and Damon both know, our mutual goal between University of Idaho and U of O is to integrate the work that we're doing at our two institutions even to another level. In the next year, we've got one grant proposal submitted and eager to work on others and bring U of I and U of O even closer together in the future. So today I'm going to talk about this balancing act between health and energy, and it's going to be a whirlwind. So hold on to your hats. It's going to be a lot of fun, but I'm going to move pretty quickly so we can get to the end and we can talk about what it is you really care about and want to talk about. So I'm just going to say that in many regards, COVID has put the bullseye on energy efficiency. And I say that because a lot of the recommendations that are coming forward to make buildings safer also use more energy. And I'll unpack that as we go, but I think that is really the theme of this talk, is to understand that we need to find a way to do either a balancing act or ideally look for synergies between health and energy and put our investment into those so that we don't create even more challenges that I hope you come away with is that this is possible. However, we need to take it seriously and invest into that synergy. And Ken, please let me know if the slides are laggy or any problems. Please interrupt me, especially. And if you see an urgent question in the chat, please feel free to interrupt me. So I'm just going to do a very, very condensed version of this piece of the presentation that Ken actually saw yesterday. And it's just trying to characterize that from the beginning of kind of human civilizations, we've always thought about health and energy. And if you'll allow me to say, we pick locations for our settlements, like along the river in Boise, for access to resources, for access to potentially energy with our hydropower facilities, to access to transportation and fresh water and fresh air and beautiful views and irrigation resources for our land, for growing for food. So to say that we are thinking for the first time about the synergies between health and energy would be completely inaccurate. But I will say this has been a push-pull over millennia, really. And I just also want to do a quick kind of flashback to what I call the 100-year flu. I would say COVID is the 100-year flu, right? We design buildings around 100-year floods, but maybe we need to start thinking about designing buildings around the 100-year flu. And here are pictures from just over 100 years ago, the influenza, or what was referred to then as the Spanish flu, and all sorts of innovative strategies to try to maintain health during this particular pandemic. You can see hospital wards outside with access to fresh air, not too dissimilar to us trying to make impromptu wards in convention centers, outdoor barbers, personal breathing zones, wearing of masks, and a really serious reconsideration of how we were occupying buildings because of a pandemic. So this is not our first rodeo. And while I think we all recognize that it is going to transform architecture, I think the question will be, well, how long and lasting are those ramifications? And reconfiguring office spaces around 6-foot circles, I think there's a lot more to it than that. And I hope we will learn something from this pandemic. And with the technology and sort of awareness that we have today as compared to 100 years ago, I really hope we'll be leveling up and talking about a new generation, really a new version of a healthy buildings future. So I'm going to try to unpack what I'll call is the unseen within the built environment. And in some cases, those are beneficial unseen characteristics. And in others, in the case of this particular pandemic, it feels like we're fighting an invisible enemy. And I really want to show that we can reveal some truths about that invisible entity, and with that knowledge and vision, be able to implement mitigation strategies to create safer spaces with a data-driven approach, as opposed to a take all the possible precautionary measures and throw that and the kitchen sink at the problem. You know, costs be darned. So first here, just to put this in context, we've been studying microbes in buildings. Well, I should say the center has, before I even got here, for a decade with the Alfred P. Sloan Foundation. But since I joined the lab 5 years ago, I've also been heavily involved in this indoor microbiome research. And perhaps you're all familiar with your gut microbes, and you understand that those are actually oftentimes really helpful for you. In fact, if you take antibiotics, your doctor might be prescribing some probiotic yogurts to help replenish them, because they are known to be supportive of human health, digestion, mood. And there's this concept called the gut-brain axis, which shows that the gut microbes might implicate mood and mental well-being and recovery from stress events, etc. And the other thing to recognize here with these sort of colors smattering everywhere is to know that we are depositing microbes into the building, and that the building is bringing microbes in and of itself, depending on its ventilation pathways and the material choices that we make when we construct the building in the first place. So what this one shows is a research project that completed just as I was coming here to Oregon. And years ago, I remember the Energy Studies Lab working with the local architects SRG on the Lillis Business Complex. It was built in 2002, and the lab had an intimate relationship. So not surprisingly, when the opportunity from the Sloan Foundation came to study microbes, we started with this building. And we understood that we started to learn that each building type had a distinct microbial signature. And as shown here, each room type within each building also has a unique microbial signature. So these are called spider plots, where we take different taxa of, in this case, bacteria, and plot them around and show how air handlers from one side of the building looks different. The microbiome looks different than air handlers from the other side of the building. And offices that are more near to one another have microbiomes that look more similar than offices that are further away. And so you take that to, well, what can we do from a building operations standpoint? Here we're showing that a mechanically ventilated ducted airway, as compared to a naturally ventilated in green outdoor and night flushed space, create really different abundances of microbes, as well as different microbial signatures. So the spider plots look different, and the outdoor microbiome had a much greater concentration of outdoor associated microbes, while the blue spider plot shows a higher concentration of human associated microbes. And that is a clue as to potential pathogenicity of the microbial community structure in those two different rooms. Now, we don't know why, and I have some hypotheses that maybe it's around the solution to pollution is dilution, and we're getting more outside air when we're delivering it through passive ventilation, especially night flush cooling, than we are in a classroom that has minimum outside air requirements when the room is unoccupied, for example. Okay, so that's the platform. Enter, February 2020 and the novel coronavirus. We sprang into action and said, look, if the biology and the Built Environment Center doesn't write a guidance document as quickly as possible, then who will? And I'm proud to say that ASHRAE Epidemic Task Force, as far as I'm aware, this was the first paper that they reviewed as they were starting to begin their guidance documents. We've had many hours of interesting conversations over the last several months, and I'll just leave that at that. But it's been a really interesting collaboration to try to bring architecture and microbiology and building science and building engineering together to support some of these guidance documents from ASHRAE as well as from AIA. Inside of this paper, we make several recommendations, but three that really stick out and are relevant to this topic today is outside air fraction, air change rate or air exchange rate, and filtration. And if you read the ASHRAE Epidemic Task Force guidance, those are three really fundamental principles. Generally speaking, the idea is the solution to pollution is dilution, and you can do that a number of ways. You can increase your outside air fraction, which is to say, instead of doing, I don't know, 80, 90% recirculated air in our buildings, you could try to increase that. And the limit you're going to run up against is your cooling capacity or your heating capacity, depending on the outdoor temperatures. Then you could increase air exchange or air change rates. So depending on the building type, you might be 1 air change rate per hour or even less. You might be in a hospital up to 25 air change rates per hour. And a lot of different buildings have different design guidelines for everything in between. And the idea here is, can you increase the air change rates? Can you get the door? Can you increase the air change rates by pushing your fans a little harder? You can't go from 1 to 20. No system can have that much of a range, even if it is a variable air volume system, unless it was designed for 20, I guess. But most you could push a little, right? So maybe you can increase your circulation a little bit. Maybe you can push the outside air fraction a little or a lot, depending on outside air temperature, kind of like economizer mode cooling. And then finally, filtration. Maybe you have MERV 10 filters in, and your system capacity and your fan pressure would allow you to go up to MERV 11 or MERV 13. You're probably not going to get to MERV 15, 16 unless your system was designed for it, and certainly not HEPA. That's probably an air handler replacement and even a ductwork replacement. But maybe you could push a little bit. So you have these 3 levers. All of them are around either the solution to pollution is dilution or filtration. And each building can probably pull those levers a little bit, but all of them cost more energy. All of them increase wear and tear. All of them require more maintenance on your cooling coils or your filters. And of course, all of them push the energy and stress the energy and probably shrink equipment life. And so we don't want to just throw everything and the kitchen sink at it. We want to try to apply the measures prudently and when we need to. And I'll pick that theme up again in a moment. Okay, so what about airborne? There's a whole host of debate between the CDC and the 100 scientists and the WHO and folks that do want to wear masks or don't want to wear masks or whatever. Just from a pure particle physics standpoint, we can say that airborne transmission is possible because we know the size of this virus. We know it can be caught up in globules that are less than 1 micron. Less than 1 micron particles can fly around the air. And there's even research to show that ultrafine particles can remain aerosolized and infectious in a perfect environment for more than 16 hours, for up to and more than 16 hours, right? And so the whole idea with this was just trying to say, look, while wherever you come down on that spectrum, from a particle physics standpoint, it's hard to deny that particles could remain aerosolized for some period of time, get caught up in air streams, and fly more than 6 feet. And that there are things that we can do about it. And here in this case, we're just simply showing 2 simple concepts, maybe 3. Increase outside air exchange, in this case through a window, exhaust air, so once through air as opposed to recirculation. And then the third being when you do introduce outside air, oftentimes, depending on the weather, you might have increased relative humidity. And there's kind of a trifecta related to relative humidity that is pretty interesting. One is that increased water content in aerosols means that those aerosols will remain, will fall out of the air more quickly. So here you can see the ultrafine aerosols are the blue, and with more humidity, though, especially those will become heavier and will fall out of the air more quickly, so you're less likely to breathe them in. The second is that mid-range humidity from 40 to 60% has been shown to be supportive of human immune function. And if you just think of this, if you've gone on an airplane, the air is really, really dry, you come out and your mucous membranes might be cracked and more likely to create structure for a pathogen to take hold and dry out the natural ciliary action that can happen as a natural preventative measure from our immune system. And then the third thing in the trifecta would be that increased humidity from 40 to 60% has been shown to more rapidly inactivate virus particles. In particular, there's a lot of research on influenza, but there is some emerging research on the novel coronavirus as well. And in general, it's fairly well accepted that mid-range humidity can more quickly inactivate influenza especially. Okay, so we've got some problems, but there are some things we can do about them, right? Well, we want to bring data to that equation. And so we had been studying with our hospital partner at Oregon Health and Sciences University for several years before the coronavirus pandemic, other pathogens. So two in particular, two bacteria, one called Pseudomonas aeruginosa and the other called Clostridioides difficile, or C. diff. Most folks have probably heard of C. diff. It's a gut bacteria that causes you to have some pretty bad diarrhea, and even in its worst case, you can have large sections of your bowels that have to be removed, your large intestine, because they become irreparable. And it's absolutely dreadful, has long-term consequences if you do survive, and it actually kills about 8% of the people who acquire it. And so we had been already studying different pathogens in the hospital environment. Insurance companies often don't make payments associated to what they determine was a hospital-acquired infection. So if they can say that they think the patient acquired it within the hospital, they can refuse payment, depending on your provider and your terms, of course. And so we pivoted in March and wrote the protocol to do the similar work on coronavirus, and we were so fortunate that the hospital agreed. And I just want to give a huge shout out to OHSU, major hospital, teaching hospital, above best practices in all of their protocols. I have to give them, props for that, and for the willingness to take on what is a pretty vulnerable experience here, and even letting me talk about it. The last thing we want to do is say that hospitals are scary places to go, because if you need to go to get healthy, you need to go. But there's also a reason why they're pushing to more outpatient procedures, and part of it is because the less time you can spend in a hospital, the less risk associated with some sort of hospital-associated infection. So the fact that they were willing to let me and let us participate and actually fully collaborate in the research is just amazing. And I think this hospital, because they're taking such a proactive approach in testing their spaces for the presence of this genetic material associated with this virus, that they're likely the safest hospital, or one of the safest hospitals to go to. And so with all of that said, here's the research. You've got two types of data on this plot, and just focus on the two empty circles on the far left. You got the black circles, which show that those are places that we used a swab, a glorified Q-tip, to collect biomass, basically dust from surfaces, even if it's invisible dust and it doesn't look like there's anything there. And then a passive settling plate, or a dish. And that's just a plastic dish that we fold open and we let it collect any particulate matter that falls in from the air for some known period of time. So really the surface swab is a time integrated signal of anything that might have deposited in that space since the last time that surface had been wiped or cleaned. And the dish is a known shorter period of time, usually just an hour or two, as we're walking around putting dishes out into multiple rooms, and then coming back an hour or two later and collecting those dishes. And here you can see that this particular room had two locations that showed the presence of the virus. And I want to take a moment just to explain that. So the presence of the virus, when I say that, what I really should say is the presence of the genetic material, or very specifically the RNA. And if you're used to hearing DNA, it's a similar concept. It's just that this particular virus is an RNA virus. So we're using that genetic signal to detect the presence of the virus. Now, that doesn't mean that the virus is still viable and intact. So some people don't like to use the word virus unless it's a viable and intact and therefore potentially infectious particle or virus. And so some people just say genetic material or RNA for something that traces the indication or the potential presence of the virus, or really the previous presence of the virus without knowing if it's still viable or infectious. Okay, so with that said, what we found here is a floor in the bathroom and the return air grill, or the exhaust air grill, where, you know, like a vacuum cleaner is sucking out all the air and the dust, there's little residual dust that we can track off of that return air grill and test for the genetic material. And we found those two spots to have the presence of the RNA. Now, what was interesting here is that this space had never had a COVID positive individual in it. And even the whole floor, or the ward that it was on, hadn't had a COVID positive individual in 11 days. And so this was another data point to say, look, we've heard that RNA decays really, really quickly and is no longer detectable. Now that may be the case, and that means there was an asymptomatic individual that may have spread virus more recently. But we are learning that this particular RNA seems to have some stability in the environment and lasts a little longer, and we're able to detect it, in this case, probably 11 days later. And the Princess cruise ship showed that they could detect the virus after the cruise ship was empty for as many as 14 days. So we have to use that knowledge to help interpret these results. The first thing that came to my mind when I saw it on the return air is, okay, we're probably dealing with, you know, viral matter, genetic material, whatever, that's in really, really small submicron particles or globules that is then getting sucked to that return airstream and showing up and being deposited on those grills. So the next room that we tested was a negative pressure isolation room. And you've probably heard about this enough in the press by now to know what it is, or if you're an HVAC engineer, you know what this is, you've designed them. Really what that means is it's slightly negatively pressured. So you're sucking a little more air out than you're supplying. And because it's an isolation room, you're exhausting that air right out of the building without putting it back through the air handler. So this is where hospitals put their sickest individuals that they're most concerned about potentially spreading a virus or another pathogen to other people within the hospital. It also means that you're bringing all the possible microbes from the whole hospital, you know, through that space because it's slightly negatively pressured. Some air might be coming under the door as quote unquote makeup air compared to the air that's supplied. Usually I've tried to balance it pretty close, and really it's about the isolation. But you do want slightly positive or slightly negative pressure so that when the door opens that nothing's sweeping out. Okay, all that said, you see 5 surface swabs that tested positive, and interestingly, the return air grill didn't. May have just been where we sampling frequency, but we did see a positive Petri dish in this case. And then finally, the operating room. And here, this is sort of the cleanest of the clean, if you will. You know, 20 to 25 air changes per hour, MERV 15 filtration. And this part of the year was quite mild. Studies were collected in April, quite mild outdoors in Portland, and we had a really high outside air fraction. So kind of the best of the best of the best. Higher than even ASHRAE recommendations for hospital settings and the new ASHRAE Epidemic Task Force guidance, which is recommending MERV 13 for commercial buildings if possible. So the fact that we saw at least the genetic material in the dust that settled into the Petri dish started asking, in this room in particular, started us asking some pretty tough questions about the possible transmission routes through air handlers. And therefore we ran this study, and this is available as a preprint. Those 4 slides that are the 3 slides I just showed you are not yet published anywhere, but these slides are. And this preprint is available on medRxiv, and it's going through peer review right now. I can explain to you why we decided to issue it as a preprint, but basically the short version is we thought it could be helpful for society. Was really exciting to see that the journal editor actually immediately forwarded it to the WHO even before it went out for peer review. So that was for us a pretty good indication of their confidence in the data as well as their belief in the importance of the work. So what we show here are 3 air handlers that we tested at the same hospital. And air handler 1 5, and 10. And you see that 4, 5, and 6 are ganged together, and 9 and 10 are ganged together, while as AHU1 operates independently. All 3 of those locations, at some point, the floors that they serve, at one point during the pandemic, had a COVID positive individual. And all 3 of the air handlers had at least the relic or the genetic material associated with the virus. It was the first paper, to our knowledge, that shows this result. And if anybody has seen any other research that's similar to this, please do forward it along, as we're trying to stay on top of these kind of things as much as we can. And this is a zoom in to each air handler location that we sampled. So we sampled 3 locations in each air handler, and we sampled 3 spots at each location. So you can see we sampled at the pre-filters, and we sampled in 3 spots. At the final filters, which is a MERV 15 filter, at 3 locations. And then after the final filter, at the air handler, or excuse me, at the supply air damper, also in 3 locations. Again, across 3 air handlers. All 3 of the locations at all 3 of the air handlers had some positive samples. And in total, all positive samples were represented about 25% of the population of all the locations that we sampled, or I should say sampling locations. So 25% of the sampling locations came back positive, but all 3 air handlers at all 3 locations within each air handler had some positive samples. And so just pause there for a second. And what that means is it was actually really interesting, because that correlated fairly strongly with the degree of positive samples from the floor level or the ward level sampling locations. And for me, that meant, wow, we could use the air handler as a rough order of magnitude surveillance strategy for the possible presence of the virus within any of the locations in the building that is served by that air handler. Basically testing that exhaust airstream, right, or the return airstream. The other thing, of course, that this suggested is the possible risk associated with airborne transmission routes or aerosol transmission routes, even and including through air handlers. So if you look at the genetic material that we collected from each of those 3 locations, this is gene copies per sampling location. And you see a high, low, high happening here. And there's lots of hypotheses that I can draw from that result. Nothing can be absolutely conclusive because, again, if you think about what really matters is there an infectious virion or virus particle making its way through and impacting the supply air damper? Impacting upon the supply air damper. We don't know that yet. We're actively working on that to determine the potential for viable virions, virus particles moving through airstreams. But we can say there was a high, low, high pattern on the pre, final, and supply air. And that suggests that we should, do additional research. Ken, I see your question about UV filtration, and I'll get to that one. Okay, so where are we headed? All of that was to say we've done some of our homework. We're learning about how microbes in general and this coronavirus specifically is moving through airstreams or moving through buildings. And what are we going to do about that? So I started off with saying, look, coronavirus is sort of putting the target on energy efficiency and saying it's going to be hard to hit some of our energy targets, our energy efficiency targets, if the big strategies related to reoccupying buildings are all about increasing energy consumption. And while that may be true, and those strategies may be important to implement for some period of time, first, we have to be careful that it doesn't become the new normal. And second, and in order for it to not become the new normal, we need to apply it strategically. We need to apply it with data. We need to use a data-driven strategy to say, look, we need to increase outside air change right now, or we need to increase outside air fraction right now, or we need to reduce occupant density in our building right now, but not necessarily tomorrow or the next day. And we want to be able to add that as another data stream to our building automation systems, as well as like our Office 360, office environment or building environmental, spatial, and sort of human resources equation. Because if the answer is just use more energy, we're going to pay a price, both in our cost of our energy as well as the potential for increased particulate matter. And interesting as it is, increased particulate matter exposure to certain populations is showing increased susceptibility to things like coronavirus. So it's a vicious cycle we want to make sure we can avoid. The other vicious cycle that I think we're all seeing play out right now is just the extra stress that this pandemic is putting onto an already difficult situation with regards to, broadly speaking, social justice concerns. And I would argue that at least a piece of it is because we're so isolated. We're isolating ourselves in so many different ways. And as a human person, it's really important for us to have community. And so if you think about that from like a 6-foot bubble and buildings, not occupying building standpoint, that's one scale. But if you could also think about that from like hey, everybody, the Wall Street Journal is asking whether urban centers are even going to be a viable thing into the future because they promote density. Well, and Boise's living this in many ways. What happens when you allow sprawl is that there's a lot of challenges associated to transportation that increases energy and particulate matter, and there's also a loss of efficiency to some extent by not being able to communicate with one another as quickly. And I would say it's impacting community and sort of health, mental health and well-being, loneliness. It's also impacting innovation. And I would say it's slowing us down right when we need to accelerate. So I'm proposing that the solution to these problems is look for the synergies between health and energy. While acknowledging that we may need to spend more energy for short periods of time, let's make sure that's an acute and isolated treatment and not a new normal. And let's make the new normal all about promoting environments that are sort of holistically supportive of human health and human immune function. And what I love when you start shifting the thinking that way is it means you can start thinking about some of our old tried and true strategies that are actually also energy efficient if we do them well, such as access to daylight, access to fresh air through windows, or passive ventilation, night flush, economizer modes. Like, yes, maybe we need to purge our buildings, but let's make sure we are taking the viral signal as a guide to purge, and that we can really ratchet up, especially during night, economizer modes when our summer nights in Boise are cool enough, those kind of things. So I'm going to unpack a couple of things in 3 categories, and I'm going to do it really quick because I want to get to Q&A. One is daylight, one is IAQ, and one is, generally speaking, control. So we know that daylight can inactivate a virus. We've known it for a long time. We've known it can inactivate bacteria for over 100 years. And there's a lot of research on UVC. And we're collaborating with a BSL-3 lab that's doing research on UVC disinfection. And it's their research, not yet published, is showing a pretty rapid inactivation. And that's UVC. UVA and UVB, there's also research out that shows by Paul Dabisch, if you look that up, D-A-B-I-S-C-H, that UVA and UVB also more rapidly inactivate the virus. So there's some hope there, I'd say, that strategy could be effective. Then there's the whole like daylighting is good. And many of you have heard me talk forever about how good daylighting is, right? Supports health, supports human circadian rhythm, which supports sleep, improves mood, improves digestion, improves alertness, and all these many attributes that can also, as a whole, support human immune function. How can we get more daylight into our buildings without the glare? This is our first spin-off company. I'm really proud to say that some of our students have spun off this company called Task Shade, and they're building micro glare protective devices that are allowing more sunlight, more daylight harvesting without the glare, and potentially more circadian dose and more microbial inactivation. Then you've got IAQ, and this is a big one. So I've already explained ASHRAE guidelines about outside air fraction, air exchange rate, and filtration. Here's one that also does an interplay with energy efficiency, and that is an old strategy, 10, 20 years old. We've talked about this, splitting your heating and cooling from your ventilation system and being able to drive each as needed. And so with a dedicated outside air system, even with heat recovery as shown here, you can either do this in new construction or retrofits, where you take over the ductwork in a retrofit or use ductwork in a new construction project for the fresh air component, and you use independent heating and cooling systems, whether that's through radiant slabs like our friends at CSHQA, or mini split ductless heat pumps, a lot of different strategies. And so that you can drive each of those independently. And then here, even at the more micro resolution scale, thinking about personalized airspaces. So each room potentially could have mini heat recovery right at the window. And these are two products that are pretty well established in Europe, one by Sagina and one by Schuko, that have basically micro ventilation and micro heat recovery right built into window units. Similar concept with Velux that has operable skylights that also admit daylight. All right, so this is a little more bleeding edge, but there's inside of these trays that our PhD student is working on, he's growing a micro fern called Azolla. And Azolla is a really good carbon dioxide consumer, and of course emits oxygen. And so we were thinking about this pre-pandemic from a create clean air spaces during wildfire smoke, but I'd say the same concept can apply to creating isolated micro airspace environments where you're less likely to share air with other individuals. And that could be done through traditional filtration. I'm just really intrigued with the kind of multiplicity of biophilia, CO2 sequestration, capture, biochar, as well as the humidification, and even perhaps intermittent UV treatment associated with some modules that could be used to grow these Azolla. We're actually thinking like a microeconomy even around this life cycle. And then finally, control, big data. Right now, we're doing data overload even in our current building automation systems. We've been working, even when I was 10 years ago, working with Idaho National Lab on the targeted energy management system tool set, basically looking for ways to use the data that our buildings are already collected more intentionally, more meaningfully, with greater impact. And I'd say the added layer of health data and human associated occupants data can bring that in particular in this pandemic to a whole nother level. But for example, luminaire level lighting controls got a whole distributed mesh network of sensors that are right now disassociated from the building automation systems that are operating our HVAC. And those two data streams could definitely do their job better together, but we need to work out the interoperability. Imagine a scenario where demand control ventilation, which is currently implemented using CO2 sensors, could instead be implemented using this distributed mesh network of occupancy sensors. CO2 sensors drift over time, they've known to fail, and they're laggy, right? Because they wait for you to exhale and wait for that air to go up into a duct and get sensed at some sensor some distance away. And it waits for the CO2 in the room to build up to the point where it's uncomfortable, muggy, sticky, and then the sensor's like, oh, I need to provide more fresh air. By that time, the meeting's over, everyone's gone. What if you could use a luminaire level lighting control signal that's real time and deliver the fresh air immediately? Okay, finally, this conceptualization of testing buildings, it's the thing I'm most excited about right now. Good Morning America picked this up, super excited. They ran it last Friday. The whole idea here is because of the research in the microbiome, in particular testing the exhaust air stream from buildings, we believe that we can put some visibility goggles on this invisible enemy, the coronavirus, and apply these strategies with greater data and therefore greater effectiveness. What we are proposing here and what we are indeed doing on campus buildings here at University of Oregon, as well as a corporate partner that Good Morning America was able to get them to say out loud their name, which they wouldn't let me air it, but they gave it to Good Morning America. So we're working on this plan with Nike right now. We're collaborating with a private sector company called EnviroTech, who's doing it in more than 60 senior care centers. And obviously we've been doing it in healthcare centers at OHSU as well as UC Davis. And what I'm really confident in is that we can detect relatively low signal of the genetic material, the presence of a virus. And with that, over time, if we're testing maybe once a day or 2 times or 3 times a week, if we see a spike in signal, then we can use that data stream to more target our intervention strategies or even our human testing. And I believe it's the way to thread the needle, get our buildings reopened, restore our economy, and do the both and. I don't care where we fall in the political debate. I'd say everybody wants to be able to reoccupy our buildings and restart our economy. And I think this is the key to do it. And I believe it's bipartisan in nature, so it has a chance to make it through. Last 2 slides. This is why I believe we can do it and why I think it's so important. The South Korean call center showed a 45% attack rate. Here is the retroactive contact tracing of that building. 45% of the people on that floor got sick. And we could have seen in the building on the floor below, the 10th floor, where most of the people got sick on the 11th, as much as, 20 some odd days before the big spike, we could have potentially spotted that person or the person on the February 25th or the group of people on February 28th. And if we could have identified any one of those and either taken the extreme measure to vacate the building or probably taken a less extreme measure, increased outside air change, increased filtration, increase air exchange rate, do targeted isolation, shut down maybe the floor 10 for 2 days, have everybody go get contact, tested or whatnot, a whole panoply of potential mitigation strategies, I really do believe we could have avoided that outbreak. And if we could have been sniffing buildings before coronavirus for pathogens like coronavirus, I think we could have avoided the pandemic, at least minimized its impact, and avoided or minimized the impact of the shutdowns. So you guys have all heard me say this before. This is the new integrated design. I love the integrated design lab name. I think it's going to stand the test of time. It'd be a great name for a long time. And it's because we keep reimagining what integrated design means. Thanks very much. Happy to chat. Happy to take the last 10 minutes for questions.
Thank you, Kevin. Just like I thought, that was even, more information than yesterday, or good supplemental information. Thank you.
I just talked faster.
I see. Well, I would like to see someone on the chat. Let's see here. You had a question. Yeah.
Yeah, Ken, did I answer your question well enough there? Ken Gallegos?
Yeah, I think so. We just had a owner that was asking about things they can implement in their building that's under construction right now to help mitigate. And it looked like there was a couple cassettes in the air handler unit that was easily retrofitted to slide a UV filter in there and just add, another layer of protection, not 100%, but something else that will work.
Yeah, I think it is another layer, and that's what we need. And, in an ideal world, that would be driven with a data stream about whether you need to use it as opposed to just blasting forever. And, there are some potential negative consequences associated with long-term use of any of these disinfection strategies, whether it's UVC or chemical disinfectants, et cetera. And that is, there's a reason it works, and that's because the pathogen, whatever it might be, has not seen it before. And the more times you reintroduce it, there's this potential for resistance. And we've seen that played out at a sort of human population level with regards to eventually antibiotics become less effective. And that is due to resistance, especially if you don't kill it completely. If you wound it a little with some exposure, but you don't zap it, then it has the ability to build up resistance either itself or from sort of replicate to replicate to replicate. When you take samples in our dish or a swab, how long does it take to get the test results? Yeah. In our lab, we're turning test results around within 48 hours if it's a research kind of project. In a partner or a collaboration that we have with EnviroTech, they're turning lab samples around in less than 24 hours. And that includes, most of the time, the shipping. So they promise 24-hour turnaround once they receive it at the facility after an overnight ship. But most of the time, they say 99% of the time, they're giving it within 24 hours of the time you drop it and the shipment happens. So it's fast enough to be actionable, in my opinion, for these kind of concepts. When you take the samples, oh, sorry, that's the same one.
Other questions, you can put them in the chat. You can go ahead and send them out, or just put a question mark in the chat. We can unmute you. You can ask your question.
Hey, Kevin. I think Shelley had one on one of her comments. She would like to know your thoughts on bipolar ionization.
Yeah, I'm reading as fast as I can on bipolar ionization or needlepoint bipolar ionization. The reading I've done so far, and I want to be careful here, like as far as an endorsement goes, I think a lot more research has been done on UVC, but it's really intriguing. Needlepoint or bipolar ionization in general is really intriguing because it appears to act on the entire airspace, not just in a duct. And I've heard of some concerns around ozone as a secondary byproduct, but I've heard other data that say it's not a concern. This is where my question goes. And then if there is an ozone source, there's always a risk of a secondary reaction associated with any of the chemicals that are already in our buildings or on our skin. And that could have sort of some chronic concerns to watch out for. So again, I'd put it into this, use it when you need it, but let's be careful of any of these strategies and using them constantly. The humidification one I think is pretty interesting. The downside to humidification, of course, is if you screw it up, then you've got risks for Legionella or any other microbial growth that you don't want. But the notion that maybe you could dehumidify less parts of the year and let the humidity indoors ride into that 40 to 60% range more often, I think that's one relatively low risk version of it. You might have some comfort issues you got to sort out. But there are a lot of companies that are promoting humidification strategies as well. And I think we have to make sure that we have a fail-safe measure. So if you are adding humidification, that if it fails to off, so to speak. And I think that's a strategy that you could explore. Humidification doesn't have huge market penetration, but it is growing. I see more questions popping in.
So there's one about, there's one about the copy of the presentation. Maybe we could add that to the website for possible download later. Maybe when you get everything finalized, because I know not all of it was published.
Yeah, it's
so
okay.
I might delay in that.
Yeah, the YouTube video is available faster probably, but yeah, I'm not opposed to sharing the slides. So if you're interested, reach out, follow up.
One question I had in regards to LLCs, you mentioned that about improving building automation response. Are you doing an experiment or thinking about maybe having occupants conduct a wellness survey? Maybe, there's Wi-Fi data integration into the LLCs. So, they could send a wellness report and where they are, what floor, what area of the building, so you can understand better.
Yeah, I think so. There's this kind of privacy question. There's an opt-in versus opt-out question. One of the things, so I think that's a great strategy to layer in. In particular relation to COVID, as opposed to sort of a broader concept of health. I think with COVID, there's such a huge asymptomatic or pre-symptomatic concern that I think that has less potential benefit because probably if people are sick, they're staying home, or hopefully they are. But I think with regards to a broader question around health, connecting, for example, access to daylight with a large scale building occupant survey is a super valuable idea. And, I think it's this additional data stream that can help guide decision-making in the future. I see Guy Newsham on. That's exciting. Guy, do you have a question?
You've got one question here from Tim Johnson also.
Oh, good. Hey, Tim.
And we're about out of time.
Pathogen sniffing, yes. Okay, pathogen sniffing in real time versus what we're doing right now as a subscription program. So right now we're sending a kit out with swabs, teaching you how to swab. You drop it in a FedEx bag and ship it back, FedEx box and ship it back. 24-hour turnaround. So everyone wants it cheaper, they want it faster, and they want it more accurate and more real time, right? So cheaper, faster, and in real time. So there are some really interesting products moving on the market. In fact, we have, at this point, I think we've got non-disclosures in place with 5 different companies, some small startups, as well as with some really large and well-established companies, all working on different aspects of this question of approaching a real-time sniffer for a pathogen. And I think it's possible, though aggressive, to see something come on the market within the next year. And I think there might be kind of this minimum viable product that's a little clunkier, and then maybe something that gets refined over time and more integrated to a building automation system. And that's my hope. And what we're trying to do is stitch together organizations that have never considered working together before. And so it's kind of the integrated design approach to this particular problem. And we'll, as soon as we have something that we can share, we certainly will. I think it's a really exciting and potentially kind of a new normal for healthy buildings.